So here is my little lecture about nature of light. So light is an electromagnetic wave. So if we have a point charge which doesn't move, around itself it produces an electric field. And if this point charge moves with constant speed, it produces still an electric field, but it also produces a magnetic field around itself, okay? But if now the point charge moves, but it changes its speed, so it accelerates and decelerates, it produces an electromagnetic wave. And what is that? It's actually the electric field and the magnetic field become coupled together, and that is an electromagnetic wave. And this wave propagates through space. And light is an electromagnetic wave. There are other types of electromagnetic waves, but light is one of them. So you can see in here a representation of the electric field, which oscillates because we change speed. We go faster and slower with the point charge. And then we have here in blue the magnetic field, and you see that they are coupled, so they oscillate in unison. And because of that, this creates this electromagnetic wave, which travels in space. So it has a direction of travel. So this is what light is. And because of this nature of light, we can represent light in different ways. So in here, you can see a representation of a wave which travels. And you can see in here that we can represent light with its wavelength, which is basically the distance between one top of the wave to the next top of the wave. This is the wavelength. So we talk a lot about the wavelength of light. This is what we mean. And the wavelength of light is often abbreviated as lambda. This is the letter that we use to represent this distance here between the two crests. But we can also represent light as the direction of travel. And this could be more like an arrow like this, showing a direction, okay? And this is called ray optics. So we can represent light this way as well. But we could also represent light as its wave fronts, which means like the parallel lines that are on top of the crest, for example. And in between the two the wave fronts, there's the wavelength. So you can see in here that we have a flat wave front, it means they are parallel. But in here, we have a bent wave front. So when we talk of optics, we will see that different elements will change the shape of the wave front. So we will talk about this. So light is an electromagnetic wave, that's what we saw, but it's also associated with energy quanta. And this energy is not evenly distributed along the wave. So we have a high probability to have condensed packet of energy, and these are called photons. So here is a full range of electromagnetic waves, and as you can see, it covers lots of things. For example, cosmic rays with very, very, very short wavelengths are also an electromagnetic wave. We have X-rays, we have UV and infrared, and then we have microwaves when you warm up your food, and we also have radio and radar and all this, so the radio waves. So all these are very, very long waves that have a wavelength of sometimes hundreds of meters. And so these are very long waves in here. So in here, in this little thing, we have visible light. And so this is what we work with really in light microscopy. It's a visible light. It's about for 400 nanometers to 700 nanometers. Before that, we have UV, and after that, we have infrared. Okay, so this is what we work with in light microscopy. So what's the difference between the short wavelengths and the long wavelengths when we talk about electromagnetic waves? So the blue side or the red side of our visible spectrum. Well, when we have short wavelengths, the light is more energetic, it's more damaging, and it's more resolving, okay? So more energies on the short wavelengths, but it is also less penetrant, so it's blocked immediately. You remember like UV, yes, it's damaging, it can damage your DNA, but it is also blocked by the first layer of the skin. It doesn't go deep into your tissues. And then if we have long wavelengths, if you think about radio waves, for example, they are really, really penetrant. So they grow across buildings. We keep hearing the radios when we are inside the building. So they are extremely penetrant, these long waves. It's the same thing when you go longer in the visible light. These are very penetrant, but they are less energetic And because of that, they do not damage our tissue so much, and they have a lower resolving power. So the wavelength of light determines how energetic, penetrant, and damaging it is, and how we perceive its colors. So you can see we have here the different wavelengths, and we have the different colors of the rainbow. And so there's a color associated with each wavelength. And also it determines the maximum resolution that we can have in the macroscopy image. Okay, so we can, now I have spoken about resolution, I have to define it. What is resolution? Resolution is a distance. It's the shortest resolvable distance between two objects, okay? So resolution is a distance, and it is never a number of pixels. And very confusing, because when you buy a TV, for example, they're going to tell you, oh, it's excellent resolution, it's so many millions of pixels. Yeah, that is not a resolution. So it's a shortcut that they're taking. The resolution is a distance. So it's a little bit confusing, because when we have high resolution, it's a short distance. So the shorter the distance we can resolve between two objects, and the higher the resolution. But you'll get used to this, because we'll talk about all this later. Right, and there is a little definition I wanted to add. What is isotropic resolution? It means we have the same resolution in X and Y and Z. So in all directions, it's then called isotropic resolution. Maybe some of you have heard this word before. So this is visible light. And let's have a look at the visible. Now we zoom into the visible light spectrum, and you can see here the different wavelengths in nanometers, 665 nanometers, and then the representation of longer waves in here as we go towards the red side of the spectrum. And now, if we shine white light on, like, for example, the sunlight or some of the light, this is a mix of wavelengths. So now we're talking about individual wavelengths, but actually white light is a mix of wavelengths, okay? And when we have a light source on our microscope, depending on the light source, we have actually different types of lights, okay? So let's have a quick look, because we always think monochromatic, but it's actually not the case. That we always say, oh, I'm exciting with the 488, but what is it that you're shining exactly on your sample? So mercury lamps, for example, it's possible. I'm not sure if anyone has a mercury lamp in their microscope this year. Does anyone know if they have a mercury lamp? You guys have a lamp on your microscope. People who would have would be using a little bit simpler wide field system would have mercury lamps. Anyone has a mercury lamp? Nobody? No, I don't think so, because I've been looking at what you guys have. I don't think you have a mercury lamp. But anyway, the mercury lamp shows the whole visible spectrum, but some of the wavelengths are much, much brighter than others. Okay, so this is the spectrum of a mercury lamp, for example. But it is white light because it's a mix of wavelengths. And then a laser is almost monochromatic. So we have only a few nanometers. So when we say the 488 nanometers, it's really maybe 1 or 2 nanometers. So it's extremely precise. Lasers are very precise in their output, in the wavelengths that they output. And then we also have LEDs. Some of you may have LEDs, and these have a relatively narrow wavelength, but it's anyway much wider than a laser. So 30 to 50 nanometers, for example. So these are the light sources that we have normally on our microscopes. So now I want to mention, yes. I want to mention several things that can happen when we shine light on our samples, on our microscopes. One of them is scattering. So in a homogeneous manner, if we have a sample that is like the same everywhere, so if in homogeneous matter, light travels without disturbance, okay? But in heterogeneous matter, which is what our samples are made of, they're made of lots of things. The particles that are larger than the wavelength, so anything that's going to be bigger than 500 nanometers, more or less, scatters light. So it means what? It means it splits the light, it disperses in all directions, okay? So scatter is in a combination of reflection, refraction, and diffraction. And I'm going to define this in a second. So it leads to a spreading of the light energy, and therefore we have a lower recorded energy and a higher haze. So we don't like scattering in microscopy. It doesn't help. And if you look at these very nice pictures of the beautiful Sarek National Park, you can see in here that we have some shrubs, but in here in this distant mountain, we cannot see the shrubs. And why is it? Because it's very likely that the shrubs are everywhere on all the mountains at the same level, but we do not see them. Why? It's because this mountain is further away from the photographer, and therefore we have between the photographer or between the camera and the mountain, there's a lot more particles in the air. There can be like droplets of water or dust or stuff like this. As you can see, as we go deeper, we have more scattering, so we lose the light. And what happens here is that we have less contrast in here and we have less color. Okay, so this is what happens as we go deeper in our sample. The light is scattered by all the bits that are in the sample. So in fluorescence microscopy, unfortunately, both the excitation light and the emission lights are scattered in the sample. So we have a double problem, let's say. And the short wavelengths encounter more particles that are larger than them. So they scatter more. And this is the reason why the long wavelengths are less disturbed. And so they are more penetrant. So it's a question of interplay between the light wavelengths and the bits that they find in the sample. So this is the reason why the long wavelengths are less disturbed when they go in the sample. So the longer wavelengths are more penetrant than the short wavelengths. So let's have a look now at absorption, because that's also something that happens, scattering and absorption. So samples also absorb light, and different materials absorb different wavelengths. So for example, when some white light comes in, lots of wavelengths comes into something, the material is going to absorb some wavelengths and then reject other wavelengths. And this is what we see. That's why we see colors of things, is because these are the wavelengths that have not been absorbed. So the wavelengths that are not absorbed escape the sample and determine the sample color. So let's see, for example, here, in the beautiful Swedish archipelago, we have the sunlight, and that is white light. It has lots of different wavelengths. And we can see in here the reflected sunlight, and that sunlight has not gone through the water. It just bounced off the water. So it's yellow. It keeps the same color, okay? But if we look here, what we're seeing is the sunlight that has gone through the water, and part of the wavelengths have been absorbed, and then some of the light escapes the water with a different color. So what we're seeing here, the color that we're seeing, blue, is because this is the color that has not been absorbed by water, so it's transmitted. So now if I would take, and I used to do that live when we had the course live, but that was a long time ago. But now if I would take a torchlight, which shines white light, and I put my hand in front of it, what you can see here is that the light that goes through my fingers is actually red, even though the light source is white. Okay? And that is because blood absorbs blue and green light. So what comes out is the rest, what's left, which is red. So it appears red. Okay? So only the red light is transmitted. In tissues, the shorter wavelengths, like blue, are absorbed faster by the tissue, and so they scatter more and they warm up the sample more. So the consequence of this for us is that if you want to image live samples, use long wavelengths, because they penetrate more, they don't warm up the sample so much, and they don't scatter so much, okay? So you should choose a long wavelength if you want to image live samples. So this was a little introduction to what light is and how relevant it is for microscopy. So light is an electromagnetic wave, and it can be represented in 3 different ways: in wavelengths, rays, and wavefronts. And the visible range we as microscopists work with is for about 400 to 700. Nowadays we work also in the near-infrared range, near-infrared. And then also shorter wavelengths are less penetrant in biological tissues, but they are more damaging and they have a higher resolving power. And so sometimes you want short wavelengths to get more resolution, sometimes you want long wavelengths to have less damage and more penetration. And scattering unfortunately acts on both excitation and emission light, so both of them scatter as we go into the sample. And then we have a loss of intensity and a loss of contrast in the image. So now I would like you to do a little quiz. So you go to Canvas, and I'm going to stop sharing. So you guys go to Canvas, and we have a little quiz there, which is very short.